So okay, so regarding the polysaccharides, okay, so as I told you, the three important polysaccharides over here. Okay, so starch, cellulose, and glycogen. So here the starch which is there, it's made up of two components as I already told you, that is amylose and amylopectin. Okay, so depending upon how many, what is the composition of the amylopectin, the solubility of the substances are depend upon that. For example, if we take starch, okay, in the starch, the amylose is about how much? 15 to 20%, whereas amylopectin is 80 to 85%. So over here, the starch, which is there, it is insoluble basically in the water unless and until you boil it and form it to a gel-like texture, it is insoluble. So this is basically what the storage form in plants. Whereas the cellulose, which is there, here this is present mainly where? In the cell wall of the plants. Okay, so the cellulose is not easily digested by the human beings because it doesn't contain that particular enzyme, cellulase, okay, by which the cellulose can be digested. So hence, it's not like easily digested in the humans or the animals, we can say. Next is what? The glycogen. So glycogen is the powerhouse for the human beings. So hence, in the animal body, whatever the storage form of carbohydrates is there, that is what? Glycogen. So this is stored in animal body. Okay, so among these three, glycogen, which is there, it's having a very high composition of amylopectin. Okay, so because of that, it is highly what? Branched as compared to the other two. Okay, so this is all about what? The carbohydrates, the hydrolysis product, what they give, linkages, and the whether they can reduce that particular solution or not. Next one is the proteins. Now, proteins. Now, what are proteins? Proteins are nothing but the biopolymers of large number of amino acid. Mainly it is alpha amino acid. Okay, so proteins are nothing but a group of amino acids. When they form a chain, then they are called as what? Proteins. So basically they are having a peptide linkage. Now, for example, how we saw in the carbohydrates, they were having a glycosidic bond or a glycosidic linkage. which joins different types of molecules together. In the same way over here, different types of amino acids are joined together by what? Peptide linkage. Peptide linkage. That is CONH. Okay, so it's having a basic structure. Okay, so amino group, carboxyl group, a hydrogen, and then you have what? An R side chain. So depending upon which type of component is going to come in the R chain, okay, the on that basis only that type of protein will be determined. For example, there are 20 different amino acids, and they all differ depending upon what their structure, their features, their characteristics, whatever it is. For example, some will be aromatic, means they may produce what? Aroma. Some may be heterocyclic, some may be simple. Okay, so like that, different types of or different groups of amino acids are there, and in that, this 20 different amino acids are being divided. Okay, now further, whatever the proteins are there, they are being classified according to their structure as well as how they look or according to the appearance. So the first one that we are going to see is the classification of proteins on the basis of their shape, that is molecular shape. Classification on the basis of molecular shape. So here we are having two types, fibrous and globular. Okay, so fibrous protein. Now, fibrous protein means what? In the name itself, it's there. Fiber-like structure it will be having. So, whatever proteins are there in that polypeptide chain, they will be like side-bys. Okay, like this side-bys, they will be present. So, here, the proteins lie side by side to form fiber-like structure. Okay, so here, whatever amino acids are there, they are joined together by certain bonds, which is nothing but hydrogen and disulfide bonds. Okay, so basically, these fibrous protein, they are present where? They are present in hair, or you can take skin, nails. Okay, so over here, you can find this fibrous proteins, and generally, these proteins are insoluble in water as well. Okay, whereas when we come to the globular proteins, globular means spherical shape these proteins will be in appearance. So hence, they will be having an intermolecular hydrogen bonding. So with this, with the help of this bond, what that particular chain will happen, or what happens to that particular protein is, they will be like spherical. Okay, like this. Okay, so with the help of this intermolecular hydrogen bonding, they will be folded into spherical structures. So hence they are called as what? Globular proteins. For example, we can take the hemoglobin. Okay, or insulin, albumin. Okay, so hemoglobin, which is present in the blood, albumin, which is present in the egg, and the insulin, okay, which is used for the sugar increase and decrease ratio. So here all which the type of protein which is present is the globular protein. So generally they are what? Soluble in water. Okay, so these are the two types depending upon what? How they appear. Now we are going to see what the structure of the protein. Now generally the structure of the protein is classified into four types. Okay, so you are having four types, that is primary, secondary, tertiary, and quaternary. Okay, so starting with the basic type of structure, which is the primary structure. So primary structure, in the name itself, it's there. It will be very simple, okay, easy to denature or easy to get, broken down. So that's why it's called as what? The primary structure. So here, basically, the whatever amino acids are there, they will be linked together with their particular sequences, but they are the most simpler form of structure. Okay, so the simple structure which can be easily denatured. Denatured means degraded. Okay. Next is the secondary structure. Also, when you go further towards the structure, it becomes a little more complicated and the denaturation process becomes a little more harder. So in the secondary structure, what happens is here in this protein, there will be long chain of amino acids. Now here in the primary structure, it will be mostly linear sequence. Linear chains will be there. So easily it can be denatured. But whereas when it comes to the secondary structure, whatever long chain of amino acids are there, they will become what? More coiled or looped or folded. Okay, hence the structure is called as secondary structure. So to keep this a little more, what do you say, coiled or looped, it is basically having two things for it or two structures in this. That is, one is called as the helical or helix structure and other one is called as the beta pleated sheath. Okay, alpha helix and beta pleated sheath. So alpha helix. Alpha helix means what? It will be like helical structure. That particular protein will be having what? Helical structure like this. Okay, so this is only called as what? Alpha helix. Okay, so as I told you, each and every chain, a polypeptide chain, is being connected by certain bonds. Okay, so that bonds make the or give the proteins what? The stability and prevent it from the denaturation process. So over here also in the alpha helix, because it's a coil twisted structure, basically it's right-handed form, right-handed turnings will be there. And here this particular alpha helix has been stabilized by what? Hydrogen bonds, which will be formed between the carbonyl oxygen of the first amino acid and the amide nitrogen of fourth amino acid. Okay, so this is regarding what? The alpha helix structure. Now, the second one is what? Beta pleated sheath. So, beta pleated sheath means what? Here, whatever the appearance of that chain is there, it will be in flat sheet like structure, like this. Okay, so it will be like sheet like structure. So, they will be running parallel to each other. Okay, so here these particular sheets are being held together by the intermolecular hydrogen bonds. Okay, then comes the tertiary structure, also called as 3D structure. tertiary structure. Okay, so here what happens is whatever the secondary structure is there, it will just twist and fold more so that it will give that particular protein a 3D structure. Okay, so here the alpha helix will bend or twist and fold to give a three-dimensional structure. This is only known as what? The 3D structure. So, like if we compare the primary and secondary structure, the tertiary structure will need more bonds to hold itself in that particular 3D structure. So hence here the number of bonds which will be involved in that linkage will be more. So here what are the bonds which will help in the maintenance of that particular 3D structure is hydrophobic bond, hydrophobic. Okay, then hydrogen bond, ionic interactions or salt bridges, disulfide, and van der Waal forces. So these many forces or bonds together gives the stability to the 3D structure of the protein. And then the last one is the quaternary structure. So quaternary structure here also again refolding will happen. Okay, so for the 3D structure, if that thing will be like this, whereas for the quaternary again more complicated will become for the quaternary structure. Okay, so here whatever the arrangement of that polypeptide chain will be more complex and the bonds involved again will be more. That is all about the quaternary structure. So as I already told you, the amino acids are the one which makes up this protein, right? So here it's very important to know the characteristics of the amino acids. Okay, so amino acids. Now what are they? Amino acids are nothing but the derivatives of carboxylic acid, which is obtained by replacing hydrogen by amino. Okay, so whenever we say amino acid, there will be two functional groups involving in it. That is carboxylic and amino. Okay, so COOH and NH2. Okay, so this amino acids, basically they are 20 in number, 20 different amino acids. And all of them are having different characteristics. Okay, so the amino acids are basically what? Crystalline, colorless, then we can say non-volatile, and also has a high melting point. So whenever the amino acids comes in contact with the aqueous solutions, it also has the capacity to act as a salt. Okay, so that is the major characteristic of the amino acid. So what happens to the amino acid actually in the aqueous state, that it starts to behave as a salt. Okay, so there is a term which is called as Zwitter ion. Okay, this is very important. Zwitter ion form of amino acid. Okay, so what happens is, as amino acids, they behave as a salt in the aqueous solution. There is something which takes place over there in the aqueous solution. So there are two groups, as I told you. One is the carboxyl group, carboxylic group, and other one is what? The amino group. So here there will be what? First of all, a carboxylic group is going to lose a proton. Okay, and the amino group will accept that particular proton and vice versa. So this is going to happen. So there reaches a point where the net negative and the positive charge, that is the in the anode side and the cathode side, it will become equal, or it will become what? Neutral. Okay, so that particular state, okay, that particular state, whenever a dipolar ion is being created, then it is called as what? Zwitter ion. Okay, so Zwitter ion means what? A dipolar ion means both positive, negative, both the charges will be present and it is neutral. So hence it's called as what? Zwitter ion form. Okay, so Zwitter ion form of amino acids means what? The amino acids behave like salts due to the presence of both acidic and basic group. So acidic is COOH and the NH2 is the basic. Okay, in the same molecule. So what happens in the aqueous solution is that particular COH group is going to lose a proton. And who will accept it? The amino group. And vice versa. So here there exists a dipolar ion. This thing is only known as what? Neutral. Okay, that is positive plus negative. And this is only called as what? Zwitter ion. Okay, so this nature is called as amphoteric behavior. Amphoteric behavior. Ampho means two. Okay, so amphoteric behavior, because they will react with both acids and bases. That is the reason. Okay, so that ion which is formed is called as a Zwitter ion. Now what happens is the pH, okay, where this dipolar ion is going to exist as a what? Neutral ion. Okay, so because it's a neutral ion, now what will happen? Neither it will go to the anode, nor it will go to the cathode. So that particular point is called as what? Isoelectric point. Isoelectric point. So what is isoelectric point? The pH at which dipolar ion exists as neutral ion and does not migrate to either electrodes, that is anode or cathode, is called as isoelectric point. Okay, so here what happens is when it's in acidic medium, okay, the carboxylic ion is going to act as what? A base. And it is going to accept the proton. Okay, so because of that, the amino acids will exist where? In the cation form. That is positive, negative. The same thing has been repeated here, just that when it becomes to the dipolar form, whatever the pH under which that particular thing is happening, that is only called as what? Isoelectric point. Okay, so this is all about the major terms which are involved in the amino acids. Now, as I told you, amino acids which forms the protein, okay, they are having the bond which is called as what? Peptide bond. Peptide bond. That is nothing but the CONH bond. Okay, So either you can say it as a bond or a linkage. So this bond will be formed between what? The carboxyl and amino group. And here there will be what? An elimination of water molecule. Okay, so the bond between which is formed between the two amino acids, that is the carboxyl group and the amino group, with the elimination of the water molecule, is called as what? A peptide linkage or a peptide bond. So when these products are formed like this by the peptide bonds, then they are called as what? Peptides. And depending upon how many peptides are present, okay, like how we saw in the carbohydrates, monosaccharide, disaccharide, in the same way here also, you have dipeptides, tripeptides, oligopeptides, and polypeptides. Okay, so depending further, whatever those amino acids are there, they are being classified depending upon how much carboxylic group is present or how much amino group is present. Okay, so for example, just write that down. Okay, so depending upon what? Depending upon the number of NH2 and COH group present in that particular amino acid, they have been divided into three types, that is neutral, acidic, and basic. Okay, so if it's neutral, means obviously equal components. So over here, whatever the COH and NH will be there, they will be same. Okay, for example, glycine, alanine, or valine. Then acidic. Okay, acidic, in this what will happen? The COH group will be more as compared to the NH2. So COH is more than NH2. Like for example, aspargine, okay, aspartic acid, so on. And basic is the opposite for the acidic. So here NH2 will be more than COH. For example,